Title of my workshop is Application of HPLC Techniques to Biological Products. Before I start, I want to know how many people work in analytical area. Anyone from an analytical R&D background? Nobody. Okay, so okay. So for those you guys, it will be too easy for you. But just I want to explain what I do. Uh, the first slide about my company. My company's name is Tara Innovations. I work as a CRO, that is Contract Research Organization. I also work as a strategic consultant, and what I do is I develop formulations. Our strength is in solving difficult problems. That's what I like to do. That's why I started the company, to solve difficult problems. We specialize in to characterize small and large molecules. So essentially, I do preform studies, I do analytical method development, and develop formulations and processes, and also packaging. The mission is to develop products and delivery systems in innovative ways using science and technology. So the whole focus is to use science and technology to develop formulations and products. So we commit to high ethical standards, and we meet challenges. We meet the changing needs of consumers. We produce intellectual property. In this case, I want to mention that I develop my own products, and I also file patents. I also make sure that if there is a chance to come up with a patent idea, then I'd certainly pass on to my clients. Before I start, I want to have two disclaimers. This slide is my favorite because there are three aspects. One is cost, time, and quality. So if somebody comes to me and say that I want to do a project at a very low cost, obviously I'm not going to spend too much time on the project, as well as the quality of the outcome will be very low. If somebody comes and says, yes, I want to finish the project in 3 months, where it takes normally 6 months or 9 months, obviously it's going to cost more because I'm going to put more people on the project. But again, the quality can be a problem. So to get good quality outcome, one has to spend enough money as well as enough time. I mean, I'm sure everyone will agree to that time and money is really essential to produce good quality outcome. And the next one is large pharma. I mean, this is for any R&D project. There are three kinds of labs. Labs in large pharma, they have a lot of funding, good scientists, good instrumentation. Obviously, they do a very good job. Academia, there are people, they are really dedicated to science. They really spend a lot of time. They produce good data, and they really do very good research. But when it comes to CRO, that is contract research, we work with the client. So the client, they are the one who decide when to stop the project or when to go on. So you can see pitfalls in my work, and it could be just because the client said, okay, this is time to move on to something else. This is I will be covering in today's workshop. First, I will give a brief background on HPLC, just to let you know what is HPLC. Then I have 4 case studies. These are the case studies from my lab. So I'll be presenting the work from my company. So when we make a formulation, we have a product, say injectable product or any tablet, but we want to make sure that the product has sufficient amount of drug in there. That's the first thing. Also, the product has to be stable over the shelf life. So this is a technique we use, HPLC. Sorry. Hold on. Okay, so HPLC stands for high performance liquid chromatography. So this is a technique we use to find out the assay value for the main drug product as well as the degradation products. So one can say that chromatography is a physical method of separation in which the components to be separated are distributed between 2 phases. One is a stationary phase and the other one is a mobile phase. So this is a typical column for any HPLC. You inject a sample, you get different bands, and when they come out, you get peaks. and these peaks correlate to the concentration of the drug in the chromatogram. So this is a pictorial diagram of how it works. So you have a stationary phase. This is a stationary phase, and these are analytes, small circles. So when you inject a sample, the mobile phase is going over the solid phase. There is an equilibrium between adsorption and desorption for analytes. So based on the binding constant for these analytes, some analytes, they have smaller binding constant, they elute faster, whereas some analytes, they bind strongly and they elute late. There are different solid phases. These are C2, C8, C18 or cyno. So it means that what's the chain length? It is, C18 obviously is more hydrophobic in nature. This is a typical layout for any HPLC. Normally we have two mobile phases, mobile phase A and B. We mix them using a gradient controller, and there is a pump which makes sure that there is a flow of mobile phase through the column. We inject the sample, it goes to the column, there is a separation, then different components, they elute at different times. There is a detector at certain wavelength, we find out the drug concentration. I just want to give you a perspective of small versus large molecules. Today's presentation is for large molecules. So what I tried to do was, I tried to have diamonds. So in this square, I have 9 diamonds equivalent to 900 molecular weight, versus this middle one has 100 diamonds, which is 10,000 molecular weight, versus 400 diamonds, 40,000 molecular weight. So here we are talking about only these big molecules, large molecules, they are very different than small molecules. Recently we published a paper in Pharmaceutical Outsourcing, and we analyzed NDAs, that is new drug applications, approved in 2014, last year. So there were 28% large molecules got approval last year, which is a big number, because in say 20 years back this number used to be maybe 1 or 2%. So clearly there is a big focus on large molecules over small molecules. So let's go back to the first term, biological products. So it is defined as virus, therapeutic serum, toxin, antitoxin, vaccine, blood components or derivatives, allergenic products or analogous products. They are applicable to prevention, treatment or cure of a disease or condition of human beings. But for a pharmaceutical scientist, normally we focus on few entities such as cytokines, growth factor, enzymes, monoclonal antibodies or fragments of monoclonal antibodies. So this is our focus as a pharmaceutical scientist. So when it comes to peptides, this is the first case study. I will be talking about analysis of a polypeptide. So when you have peptide, there are several amino acids. They are attached to each other through peptide bonds. So you get a straight chain, and in proteins they fold to produce a 3D structure. So to show it further, you have a straight chain primary structure of polypeptide. It can form alpha helix or beta helical structure. It can also form layering, because there is intra and intermolecular bonding. So in this case there is intramolecular bonding, this is intermolecular bonding to form layers. These molecules, they also form tertiary structures, so that these helical structures they fold and to form these tertiary structures. Some of these they combine to form a quaternary structure. When we want to develop an assay, what we do is we meet with the client and we come up with the strategy how to develop an assay. So in this case, for polypeptide, normally we always use C18 column. These are C18 columns are used normally for HPLC. We tried several columns, and we got this chromatogram. So normally in chromatography we look for a very sharp peak. This peak is kind of very broad. So we need to have a very sharp peak, very tall peak, very skinny peak. Also, we want to have a good resolution. So as you can see, these two peaks are overlapping. So this is not a good chromatography. I mean, it's not so bad, but it's not very good. So what we did was we tried to use a new type of HPLC column. So this column, it's called Kinetics XB C18. So this is from company called Phenomenix, and in general, all these particles are packed in a column, and in most stationary phases, these are porous materials which are porous through and throughout. So what happens is when you pass the analyte, it goes inside the particle and it comes out after some time. So the company came up with an idea of having solid core and then coated with a porous coating. So it means that the molecules, they only penetrate up to a certain level and they come out faster. So this way they can improve the resolution of peaks in the chromatogram. Here they have, for this particular column, they have this isobutyl octadecyl silyl side chains. So this is a side chain. It helps to bind or absorb the drug molecules. There are different groups like TMS, that is trimethylsilyl. They are inert substances. They prevent secondary interaction with the particles. So with this column, we got better chromatography. We have nice separation. The peaks are sharper than before. So we thought it's a better than what we found with just normal C18 column. Again, this project is still going on. Just to give you some idea, what is the problem? One thing we do is we test different parameters. So one is to check the effect of pH. So what happened was when we injected mobile phase with different pHs, we found that the peak area is changing. So as you know, polypeptides, these they have different ions or different charge on their molecules. So if in that case, I think what is happening is as you change the pH, the ionization changes, and so also the absorptivity. So we need to still work on this assay, and it's not done yet. The next three case studies are based on the same antibody. So I wanted to go over what is the basics of antibody. So this is the peptide, very small molecule, and this is the IgG antibody. As you see, the molecular weight is just 1 kilodalton versus 150. So obviously this is a huge molecule compared to a small peptide. We also look at FAB. These are the fractions of the antibody or the mini body. So as you can see, the molecularity is different. So it sometimes helps to develop the drug delivery system. If you get into details of antibody structure, they have mainly two bands, heavy chain and light chain. These chains are they are bound together through disulfide bonds. Again, you can see disulfide bonds between heavy chains too. In this case, we have variable region as well as constant regions. So when you have in the same isotypes, these regions are constant. That's why they are called constant regions. The antigen, it binds to the top portion. So this is the most important region for any antibody. Normally, you have NH2 groups, that is amino groups at this end, and carboxylic acid groups on this end as well as at this end. This is called FAB fraction and this is called FC fraction. So if you go further into antibody structure, you can see that it's very complicated. This is a light chain, and as you see, it has a really a very complex 3D structure, as well as even heavy chain has the same kind of structure. You will see bonds within the heavy chain as well as between heavy chain and light chain. So overall, if you consider antibody, the antibody will have different things. One is the total volume of antibody, then the charge on the antibody, as well as hydrophobic regions on the antibody. So when you develop a product with any antibody, you have to look at these different properties of the antibody. So the second case study is using SEC, that is size electron chromatography, for the analysis of the antibody. So ACC is a chromatographic method in which the analyte molecules are separated by their size. So the key requirement is, in this case, the analyte doesn't adsorb on the solid phase. It just goes to the pores, and the large molecules which are larger, because they don't go in the small pores, they come out faster or they elute faster. So the normal measure of volume is hydrodynamic volume. That's what people consider when it comes to finding the size of the molecule. There are different ACC columns based on the molecular weight range. As we have seen, the molecular weight can range from 1 kilodalton to say 600 kilodaltons. So based on the range it can cover, there are several different types of ACC columns. The rule of thumb is, as you increase the column length, you will increase the resolution, that is separation of peaks. As you increase the column diameter, you increase the capacity of the column, how much you can load on the column. So this is the way ACC works, that you inject a sample in the column. You are running a mobile phase, so you have a mixture of 2-3 analytes in the sample. This is a cross section. You can see this is very porous. So as you pass the mobile phase, the large molecules, they don't get into pores, they start moving faster. So in this slide you will see these large molecules, they have moved faster, they form a band. Small molecules are they are at the other end, and as you pass more mobile phase, at one point the large molecules, they form a peak, they elute, and you get a peak in the chromatogram. After some time you get small molecules eluting and you get a second peak. That's how you separate different molecules using SEC. So this is the system we used. We have HPLC pump, Waters Alliance 2695, detector was Waters 2487, and the molecular weight of the antibody used is 100 to 150. Because it's somebody else's project, I cannot give you specific molecular weight of the antibody. The concentration we used was 5 mg per mil. Normally we injected antibody at 1 mg per mil concentration. For antibodies, it is very important to use the right diluent, because antibodies, they tend to denature if you don't use the right concentration of buffer, as well as pH, as well as salt. So it is very important to find out upfront what diluent should be used. No, we cannot just dilute with plain water. So this is the condition we use for our SEC method. So we have YMC pack diol 200 column, which is a big column, 30 centimeter long. The diameter is pretty big. Normally for HPLC we use columns 4.6 or lower diameter. The mobile phase we used was 50 millimole phosphate buffer with 150 millimoles of sodium chloride, pH 7.2, and it was at room temperature. The flow rate is always low. For ACC, normally we use low flow rates. For in other HPLCs we can go 1 to 1.5 mL per minute. The run time was 30 minutes, pretty reasonable, and the detection was done at 220 nanometers. Actually, one thing to mention was, whenever you use large molecules, you have to use sodium azide. You want to make sure that there are no bacteria in the system. That's very important for ACC. So this is the chromatogram we obtained for our sample. So this is the main peak, and because it's ACC, it means that large molecules, they are eluting faster. So obviously this has to be an agglomerate. When it comes to antibodies, this is it's called monomer, and they can combine to form dimer, tetramer, like that. So this must be some kind of aggregate with the monomer. In any HPLC, we study different parameters. The first parameter is flow rate. So this is the flow rate we started with 0.5 mL per minute. So we wanted to see the effect of lower as well as higher flow rate. And as you expect, when you decrease the flow rate, the retention time increases. So we have 17 minutes instead of 13.6 minutes, and when you have faster rate, 11.3 minutes for the aggregate. Other parameter to look at is salt concentration. We started with 150 millimoles. So we wanted to see the effect of 125 versus 175. Here we did not see any effect of salt concentration. The other one is effect of pH of the mobile phase. So we started with 7.2, we wanted to go down to 7 and 7.4. Again, there was no difference or no effect of pH on the analysis of this antibody. So before we started using this method for our sample, we thought let's find out how effective, how functional it is. So we can, we get a mixture of different large molecules from biorad. So we got a mixture in a gel, we made a solution out of this. So this mixture had thyroglobulin molecular weight 670 kilodalton, then gamma globulin 158 kilodalton, ovalbumin 44, myoglobulin 17, and a small vitamin B12. So when we injected on our system, it gave a nice separation. So as you can see, these molecules are nicely separated. The biggest molecule, thyroglobulin, it came out fastest. The retention time of our drug was about 15 minutes, and so also the RT value of globulin. If you see this, the globulin, this is about 158 kilodaltons. So that was our molecular weight of our antibody. So we thought, yes, our system works pretty good. We tested the effect of temperature on the stability of the compound, and there was no effect of temperature. We also looked at the effect of chloramine T, that is used for oxidation products. So it's an oxidizing agent. So again, we did not see any effect of that. So either the compound is stable for oxidation, or maybe the system is not suitable for oxidative deck products. When we looked at the effect of pH, that is the basic pH, in this case the basic pH 10.32, the peak area was constant. So it shows that there was no effect of pH or the basic pH on the stability of this antibody. When it went to acidic pH, you can see the peak area decreased tremendously, and so also the percent peak area. It means that the compound degraded with acidic pH, and we produce a lot of degradation products. We tested effect of higher pH. Instead of 1.2, we tested effect of 2.7 and 3.4. What we found was the peak area was same, but then the peak shape, the height decreased, as well as it showed tailing. So it means that the compound was forming smaller molecules, but they were co-eluting with the main drug. But overall, again, at this time, my client said we are happy with the method. So the conclusion is we thought this SCC method was good enough for the antibody AB, and also the method is stability indicating. Now this term is very critical for any pharmaceutical chemist. The method you develop has to be stability indicating. Let's move on to the next case study. This is the title is hydrophobic interaction chromatography. It is called HIC. So in this case, the chromatographic technique that separates components based on interaction of hydrophobic groups on the molecule with the hydrophobic ligands on the media. So essentially there is interaction between the solid phase and the analyte. So here we use ProPack HIC column, which is a smaller column, much smaller, 10 centimeter column, and 2.1 millimeter diameter. The column was purchased from Thermo Scientific. This shows how what is the mechanism. So if you see this, we have a surface and with hydrophobic molecules. Then there is a mobile phase. In this case, in the mobile phase, the water is disordered completely. So it is randomly distributed, except there is a region here next to these molecules which is ordered water. So also next to the analyte, if you see this, there is a region where the water is ordered. So it means that the water molecules are tightly packed nicely. So this is important because this water is not available for diffusion, and this water, it is available for diffusion in general. For HIC, the rule of thumb is if you have low salt, there is less interaction between the analyte and the hydrophobic chains. But when you have high salt, there is interaction between the analyte and the hydrophobic chain. So this is the fundamental of HIC, that under high salt concentration you see binding, under low salt concentration you don't see binding. So when you develop a method, you have to have a lot of variables. So in this case, I want to go back to this slide, because this is what we saw, that you have low salt, no binding, high salt, high binding. So one mobile phase, it had low salt concentration, the other mobile phase had high salt concentration. But we also tried to use mobile phase C with just plain water. So we thought maybe we can also change the grade, change the composition during analysis. Also, there is I had to explain to. Let me go to this slide first. So when you do chromatography, you start with certain compositions. Say, for example, 90% of A and 10% of B. With time, you change the composition. Say 50% of A, 50% of B. So the change is either linear, or it could be non-linear. So based on the analysis, you have to try different things and see which one works the best. So this slope of gradient is always change, and you find out empirically what works for your system. Also, you need to know when to change. So it's not that nothing is fixed. So everything is variable. It's all based on your molecule. Also, we try normally different flow rates, as you have seen before. Here we use ammonium sulfate as the salt to increase the ionic strength. So we also tried different concentrations, 400 millimolars, 500, 750 millimolars. The temperature effect on the column, temperature of the column is very critical. Normally for HIC, as you increase the temperature, you see more retention of the molecules, so more binding at higher temperature. So this is also a very important variable for HIC. So this is the actually these are the conditions we use for HPLC analysis. So we have ProPack HIC column, and then we use phosphate buffer with 250 milligram or 0.05% of sodium azide. Mobile phase B was with ammonium sulfate at a high salt concentration. Then we, instead of water, we use ACN as mobile phase C, and the wavelength of detection was 280 nanometers. The temperature was high, 40 degrees. So this is a typical gradient table. So you start with 98% of B and 2% of A as a mobile phase. Then at 10 minutes you have 47% of B and 50% of A and 3% of ACN. So in this case you can see that there is a gradient from 98 to 47%. So almost in 9 minutes you get almost about 51% change in mobile phase B. So the slope can be about 6% per minute. In this here, from 10 to 20 minutes, it changed from 47 to 37%. So only 1% change in slope in this section. So this is what you do. You come up with a method which works for you. So this is a chromatogram which we felt it was satisfactory. The peak shape was okay. I mean, I worked with small molecules. So in small molecules, this peak shape is really, really tall and very slim. Almost, I would say only so much. So with small molecules, you get really good chromatography, and you don't expect really same kind of chromatography with large molecules. One aspect to look at is the linearity. So if you plot concentrations versus peak area, you get nice linearity up to 1 mix per mil. In this case, we could study the oxidation of the antibody with chloramine T. So if you see that when we expose to different amounts of chloramine T, the percent peak area, the percent main peak decrease. So obviously chloramine T is oxidizing the molecule, the antibody. We also try to stop the oxidation reaction with sodium bisulfite. So it does stop. So it did stop the reaction of chloramine T. But then we exposed to only sodium bisulfite, it did not produce reduction of the molecule. So the molecule undergoes oxidation but not reduction. When we expose to acid and bases, so as you you can see the peak area with acid it decreased tremendously, whereas with base it did not. That's what we found with ACC2. So the molecule undergoes degradation in acidic condition. Now the last case study is ion exchange chromatography. For all the proteins and peptides, there are positive or negative charges, but because they fold and unfold, the net charge can be either positive, negative, or there may not be any charge at a specific pH. So in ion exchange chromatography, molecules are separated based on the charge they have on their surface. I found that this method is most complex because molecules they ionize to different extent at different pHs. So the variables are, we can change the pH of the mobile phase A, pH of mobile phase B. We can also use a third component, either ACN or water as mobile phase C. Then there was an effect of also salt concentration on the analysis, and also the temperature of the column. So we use a Dionex Propack WCX10 column. So this 10 stands for actually 10 microns. This is a typo, it should be 10 microns, non-porous ethyl vinyl benzene column beads. So on this column there is also hydrophilic layer of some polymer which prevents secondary interactions. So this column is for cation exchange chromatography. It means that the charge on the solid phase is negative, and all the cations they get attached to the solid phase. So this is how the system works. That you again, as I said, this is very complex, so I need to explain to this. So you start with this is a column. You inject the mixture of analytes. So you have the solid phase. In this case, this is for anion exchange chromatography. It means that the solid phase has positive charge, and these are surrounded by buffer counter ions. So you start like this. So when you start the mobile phase, so the analytes they come into contact with solid phase. They replace the buffer counter ions, so they get absorbed or they attach to the solid phase, and the buffer ions they elute. So after some time, when you start, this is the next phase, desorption phase. So you start injecting the salt, and salt will replace the analyte slowly. So based on the binding of each analyte, they are released separately. So you get the triangles first and the squares, and then you have solid phase surrounded by the salt, the one you are injecting, and then you have to regenerate again the solid phase. So this is like a water softener system we use at home. So it is exactly like that. So to go back, this is the system we used. We use a Propack WCX10 column. The mobile phase, again here I want to specifically mention that the pH of the mobile phase A was only 5.5 with sodium azide, and the pH of mobile phase B was 7.5. So you can see that there is a pH gradient as well as there is a salt. So in mobile phase B we have 200 millimoles of sodium chloride. So you change the pH as well as you change the salt concentration, and the runtime is 60 minutes. So this is very long normally, because when you have 60-minute runtime means in 10 hours you can get only 10 analysis done. So it's normally nobody likes to have such a long runtime. This is a gradient table we came up with. So you have a mobile phase A, it has no salt concentration, and so you start with this. The analytes they bind to the stationary phase, and as you increase mobile phase B concentration, the salt in the mobile B, it will replace the analytes and they will start eluting out. So the chromatography is not as good as one in HIC. So you have 3 fractions. So this is a main peak, and then you have 3 other fractions, 3 other deck products. When we use chloramine T to understand the oxidation, you can see that the compound, the retention time of this was 21.22 minutes. So if you see this, the peak has disappeared from here, and these are the deck products. So the compound produce, it oxidize and it produces degradation products which bind less to the column. So, but in general, I would say the method is good enough. But again, at this point, the client said, we are happy with the method, so we had to move on. So we stopped our work. So this is my last slide. Overall, HPLC is a very powerful technique, and small molecules, it just works wonderful. For large molecules, things are very difficult. But then again, there are a lot of new columns available these days, so one must try different new columns to come up with a nice method. And again, thank you very much for your time.